All right, so chapter 16 is kind of skipping a few chapters. We're going straight into biochemistry. We really don't have a lot of time to go into biochemistry, but if you take Chem 131, you'll go into it in a lot more detail. So let's kind of break down biochemistry into its most basic components. So biochemistry is the chemistry of living things. And their processes. So one phrase I vividly remember from when I was in college was a teacher telling me, biology is really chemistry, chemistry is really physics, and physics is really math, right? So everything's super related, and there's this whole branch of chemistry that's focused on biology, right? And so there's a bunch of molecules that we refer to as biomolecules, meaning that they're important in living organisms and their processes, right? So does anybody know a major class of biomolecules? Start throwing names out. Proteins, that's a big one. So what are proteins used for? Building stuff, right? So they help build major cellular compartments. They're also important in like building muscle, right? So if you work out, you probably take protein powder or eat a high-protein diet. All right, what's another major biomolecule? Carbohydrates. So those are pretty big. Does anybody know another name for carbohydrates? Or sugars, right? So carbohydrates are really just fancy words for sugars. So proteins, carbohydrates, those are two big ones. What's another one? Lipids. So lipids are fats. So those are the big energy ones that we use, right? So proteins, carbohydrates, and lipids we can all get energy from. There's also a fourth one that's used for storing genetic information. Does anybody know what that is? Nucleic acids, right? So those are found in DNA and RNA. So I'm sure a lot of you guys have seen these in your biology courses in high school or even here at TCC. But we're going to go through these in a little bit of detail, but not an overwhelming amount of detail. So let's jump into carbohydrates first. So carbohydrates, we said, is really just a sugar. And there's a bunch of them. There's monosaccharides. What do you think a monosaccharide is? It's one sugar, right? Pretty easy. So one sugar unit. So something like glucose would be considered a monosaccharide, and I'll show you the common monosaccharides here in a second. There are also disaccharides. Using common sense, what are these? Two sugars. So it's kind of like a polymer, but you only have two chains linked together, right? All right, and then the next ones are just straight polymers. And these are sugar polymers. So let's start with monosaccharides. I said glucose is the main one that most of you have studied in biology, right? So glucose is produced in photosynthesis. It's one of our main energy sources as eukaryotic organisms. However, there are some other ones, right? So glucose is shown on the left here. Galactose looks pretty dang similar to glucose. Can anybody spot the difference? Yeah, it's highlighted in green here. So really the only difference is this OH on galactose is pointed up, or the OH on that carbon in glucose is pointed down. So they vary in one of their carbons. It's kind of flipped a little bit. So they're very similar. They have the exact same molecular formula, but they're arranged differently in three-dimensional space. Does anybody know what we call a molecule with the same molecular formula but different arrangement? Isomers, right? So these are isomers of one another. So they're easily confused because of that. Fructose is another one. So fructose is arranged a little bit differently. It's actually a 5-membered ring rather than a 6-membered ring. Fructose is really interesting. It's been linked to a lot of metabolic problems. So if you've heard about high fructose corn syrup, there's a lot of speculation that if you have too much fructose in your diet, it kind of messes up your metabolism. However, fructose is found in table sugar, and we'll talk about that in a second. So these are the three main ones. I don't expect you to memorize these, but what I do want you guys to be able to do is differentiate between monosaccharides, disaccharides, and polysaccharides. All right? The other thing, too, you may notice is in all of these, we've got these groups kind of pointed up. So anytime you see these pointed up, at least in this drawing representation, these are all referred to as D sugars. So D sugars always have those pointed up. D sugars are the natural sugars. The interesting thing is chemists can make them where they're pointed down. So flip them in the other orientation, and we can make something like L-glucose. The crazy thing with L-glucose is it tastes just like sugar. It tastes just fine, actually. However, it's 0 calories. Problem is it's so expensive to produce, it's just not economical to put that in as a low-calorie sweetener. So D sugars always have that specific orientation. All right, so let's talk about disaccharides next. They are linked via glycosidic bonds, right? So a glycosidic bond just means a bond between two sugars. So if we tried to identify the glycosidic bond, where do you think it would be for this one? It would be that oxygen in between, right? So over here, this would be our glycosidic bond. If we look at sucrose, we would have this one, and lactose is arranged a little bit differently, right? So let's take a look at the one on the far left first. This is maltose. And maltose is actually 2 glucose units. Does anybody know what maltose is found in? Malted barley, right? So it's commonly used for fermenting. So beer, you take malted barley and you ferment it. Sucrose is the one right next to it. Sucrose, instead of having 2 glucoses, has a glucose on one side. So over here, let me circle it for you guys. This one on the left-hand side is glucose. Does anybody recognize the one on the right, the 5-membered ring? Fructose. So sucrose is common table sugar, and it's a glucose-fructose disaccharide. So this is the glucose. And then over here we have fructose. This is also known as just common table sugar. That's one misconception a lot of people have, is they think of regular table sugar as being glucose. Not usually, unless you're buying a specialized sugar. Common table sugar is sucrose, so it's a disaccharide. All right, the next one that we have, again, has 2 sugars linked together. This one on the left-hand side is galactose. That's that isomer of glucose. And then over here on the right-hand side, we've got glucose. So again, it's a disaccharide. What's lactose found in? Milk, right? So some people are lactose intolerant, right? That means they have a hard time breaking that glycosidic bond into free sugars. So this is often referred to as milk sugar. One thing you may notice is that lactose, that glycosidic bond highlighted in green, is oriented a little bit differently than in maltose and sucrose. If you go on in biochemistry, you'll talk about it, but there are actually different linkages called alpha linkage and beta linkage. We're not really going to get into that, but your body has a really hard time with that linkage that's alpha, so it's kind of sticking up like that rather than down. It has a harder time digesting.
Doesn't your body stop producing that enzyme to break that down by the time you're 2, unless you continue to say drink?
Yeah, it's really interesting. So, side story. I went to China, and apparently in China, people stop drinking milk at a pretty young age, which means pretty much everybody in China is lactose intolerant. And you're correct. Once you stop having milk, you develop lactose intolerance because your body forgets how to metabolize milk sugar. Yeah, it's an interesting problem. You can buy lactose-free milk, and you probably have seen that in the grocery store. All right, so those are the disaccharides. Let's go into the polysaccharides next. These were our sugar polymers, right? So if we look at this one on the top is cellulose. Does anybody know what cellulose is found in? Plants, right? So this is our polysaccharide found in plants. It's primarily used for cell wall structure, right? So it provides support to plants. It's that woody stuff typically you think of. And if we look at this, really all it is a glucose, 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 and so on. So all of these are just glucose monomers that are again linked together through this glycosidic bond. And the glycosidic bond you can see is right here, 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 and here. And if you notice too, like with lactose, I said that this bond in lactose, specifically right here, is kind of pointed up, and I said people have a hard time digesting that. Let's go back and look at cellulose. You notice how that glycosidic bond's kind of pointed up? So again, these are alpha linkages. Can we eat cellulose very readily? No. Or sorry, this is a beta linkage. Sorry, I'm getting mixed up. So beta linkages you have a hard time digesting. And because cellulose has this beta linkage, we have a very, very hard time metabolizing it. So humans can't metabolize or let's just change this, digest. You can metabolize it sometime. Does anybody know how some animals get around this? What do you think?
I just know dogs never really want to grow up,
They eat plants, yeah, so it can make you sick. Does anybody know how like cows eat grass, which is mostly cellulose? What do cows have that's special? Yeah, 4 stomachs, right? Does anybody know what rabbits do? It's way grosser than a cow. They eat their poop. So rabbits are referred to as hindgut fermenters. So they eat a bunch of cellulose, poop it out. The bacteria in their poop breaks down all of the glycosidic bonds into free sugars, and then they eat their poop. So mammals are pretty weird with adapting to that problem of digesting beta-linked sugars. So instead of that, we try to find sugars that are more readily accessible than cellulose. Yep?
So is that version of glucose, since it's shaped weird, is that just for drawing? Or is that
That's just for drawing. That's referred to as a chair conformation. If you go on and take organic chemistry, it'll cover that. Yep.
That little sweet line means it just keeps going on.
Yeah, exactly. So this is a big, long, infinite chain, theoretically. So they get tired and they use that instead of brackets. All right, what's the polysaccharide that we eat if we can't eat sugars? Think of potatoes. We eat potatoes, right? What's in that? Starch. So that leads us to our next one. This is amylose, which is a starch derivative. Amylose is one big long linear chain. Starch is actually branched a little bit, so it's not one long chain, it's kind of a branched chain. But if you notice, all of these are linked together where these are pointed down. And so these are referred to as alpha linkages, and these are much, much easier to metabolize. In both of these, meaning both cellulose and amylose, you still have just repeating glucose units, but they're arranged differently. So let's make a little note over here that alpha linked sugars are easier to digest. These are commonly found in what you think of as starchy foods, right? So potatoes being the prime example. Does that make sense? All right, there's one last one. If you've taken biology, you store a lot of carbohydrates in your liver. Does anybody remember that polysaccharide? This is like our double jeopardy question. It's called glycogen. So your body will store a bunch of polysaccharides in your liver known as glycogen. They're actually really similar to starches, but they're super duper branched. They're still alpha linked, but this is an example of what is stored in your liver. Typically, if you have some sort of metabolic disorder like diabetes, your body's not processing carbohydrates correctly, and that's when you have to monitor your free blood sugar. So I'm sure many of you guys know somebody with type 1 or type 2 diabetes that has to do that. That basically has to do with an imbalance of storing carbohydrates in your liver and releasing them back out. So even with this glycogen, it's still a glucose polymer. It's just highly branched. Does that make sense? All right, so those were the three main classes of carbohydrates. I know we're going through this pretty quickly, but I did want to jump into lipids really quick, specifically because we're going to be using them in lab tomorrow. So we said that lipids are really just fats, right? So if you have a molecule, how can you really quickly, easily tell at home whether or not it's a fat? Does anybody know? Let's say you've got a kitchen sink. You can't wash it off. What will it do if you put it in water? Typically separates out. It doesn't mix with water. So that's the easiest way to spot a lipid. Sugar will dissolve in water, right? So that's not a great way of testing for sugar, but fats typically will float, or if they're really dense fat, they'll sink. So they're classified by being insoluble in water. And why do you think they're insoluble in water? They're hydrophobic. What sort of intermolecular force do they likely have? So if they had H bonding, they might be soluble in water. So what do you think they're mostly composed of? The nonpolar ones? What are the nonpolar ones? Remember gecko feet? London dispersion. So they have large hydrophobic regions with London dispersion forces, which makes sense that they don't dissolve in water, right? So like dissolves like. London dispersion is really different than H bonding, so they don't mix well. So let's take a look at how fats are kind of made up, at least your typical triglycerides. And your typical triglyceride has two main components. The first one is glycerol. And with glycerol, you've got these three alcohol groups coming off. And then the other portion that you have contains this unique functional group. What functional group is in that molecule on the right? Close. So a ketone would have carbons on both sides. Which one has an OH coming off that carbonyl? Carboxylic acid. So this is called a fatty acid. It's a carboxylic acid derivative. And why do you think it's called fatty? Because it's got a super long nonpolar CH chain coming off the end, right? So it doesn't behave like a typical carboxylic acid because it's got this super big nonpolar region. So long nonpolar tail. And we said it's got this carboxylic acid group. And over here we've got glycerol. And if you guys remember, yesterday we talked about condensation polymers, right? So what do you think we can kick off if we merge these together? Anybody know? Water, right? So if you think about this, really all we're going to do is just do a condensation reaction between these and kick off water in the process and then link them together. So let's take a look at what happens when you link glycerol with 3 fatty acids. Oh. We formed a new bond. And what functional group have we created? It's no longer a carboxylic acid. Oh. Does anybody know? Ester, exactly. So now we've created a new ester linkage. So typical triglycerides have ester linkages in them. And then tomorrow what we're going to be doing in lab is we're actually going to be doing the reverse reaction, which is kind of weird to think about. So tomorrow we're going to do this reaction going backwards, take a triglyceride or a fat and break it into the individual fatty acids. And then when you deprotonate those fatty acids, meaning you remove the proton from it, it becomes soap. So really soap making and fat making are just reverse processes of one another. So they're closely related. So this backwards reaction is referred to as saponification. It's pretty interesting. How many people have seen Fight Club here? Only a few people. So Fight Club, they talk about soap making. Does anybody remember the fat that they use in Fight Club? It's pretty gross. They go out and find human fat. So a pretty weird movie. It's a fun movie to watch, though, and they actually got the chemistry pretty correct in there. It's one of my favorite movies. All right, so let's talk about typical fats in your diet. So you may notice on nutrition labels 2 things listed: saturated fats and unsaturated fats. So if you think of a saturated fat, is it typically a solid or liquid? Anybody know? It's a solid. So if you think about like cooking bacon, right? The gunk left over after you cook bacon is high in saturated fats, and that's bacon grease. So it's typically a solid fat. Where unsaturated fats are typically liquid. What's an example of a unsaturated fat that you might use in the kitchen? Olive oil, right? So olive oil is typically unsaturated, meaning it's typically going to be a liquid, right? Unless you cool it down really cold. All right, so now we've got to kind of decipher what makes a saturated fat different from an unsaturated fat. And we talked about this briefly in our organic chemistry section. So do you guys remember what saturated means? Full of hydrogens, right? So it's typically an alkane, exactly. So saturated fats have all CC bonds. Unsaturated fats have one or more carbon-carbon bond. And if you think about unsaturated fats that are natural, is that carbon-carbon bond going to be cis or trans? Which one's bad for you? Trans fats are bad for you, so cis fats are the normal ones. So typically, these will be cis alkenes that occur in unsaturated fats. Depending on your food label, too, you may even have more details in this. You may see things like mono unsaturated. What does mono unsaturated mean? One alkene bond, not more than one. So that just means one CC bond. Depending on the placement of those double bonds, they have different health properties. So I'm sure a lot of you have heard about omega-3 fatty acids. That really has to do with the positioning of the double bonds in unsaturated fats. So some unsaturated fats are really good for your diet. Other ones you typically want to avoid. So be aware of that. If you get a chance to take a nutrition class here, they're really fascinating to learn about. There's a whole bunch of them that are listed in this table in your book. So it's pretty interesting to go through these. The first one listed is butyric acid. That's found in butter. And then there's caproic acid. That's also found in butter. You can tell those are shorter ones. They only have 4 and 6 carbons respectively. So they typically have a much lower melting point. And then once we get into the longer ones that are in coconut oil, the boiling point goes up because we have more carbons. And then palm kernel oils, even higher. And then oil of nutmeg is trimyristin or myristic acid. That has a very high melting point. And then you can go all the way up into beef, which is got a higher boiling point than all of the plant ones. And then you can run into the oils that are found in olive oil, soybean oil, fish oil, and so on. But the big difference I want you guys to know is the difference between saturated and unsaturated fats. Those are the most essential ones. Does that make sense? Okay, there's one other type of lipid that's also very important. Or actually two. I'm forgetting one. So the first one is waxes. So if you think of insects typically have some sort of waxy coating on their exoskeleton. What's the purpose of the waxy coating? Anybody know? It makes you waterproof, right? So if you cover yourself in oil, water's just going to beat off of you and you won't drown as readily. The coolest thing is watching some of the insects that float on water. The reason they're able to float on water is this waxy coating on their feet that makes them really, really hydrophobic. Hydrophobic enough where they actually float on top. So if you ever get a chance to go to a lake around here, chances are you'll see water skippers skipping across the water due to that waxy coating on their feet. And waxes are a little bit different than your normal triglycerides. And I'll show you guys how they're different. What functional group is in here? It's another ester, right? But unlike a triglyceride, you only have one ester linkage, not many of them. So with a triglyceride, we had 3. This one only has 1. And then off of each end, you've got a nonpolar region and nonpolar. This used to be a really important industry. If any of you have read Moby Dick, they used to go out and hunt whales for the spermaceti wax inside their heads. And then they would use it for lamp oils and other industrial uses. However, nowadays, most of the waxes that we get are created synthetically. So we don't go out and hunt whales for their wax anymore. All right, there's one other one that's also really important in biology, and this is cholesterol. And I'm not going to ask you guys to draw this, but you can see it's pretty complicated, right? What's the purpose of cholesterol in cells? Does anybody know? From bio? I'll give you a hint. Cholesterol goes into cell membranes, kind of embeds itself in that lipid bilayer. Nobody?
So it's keeping like a balance.
Yeah, it's to help maintain a certain rigidity to that cell wall. If you don't have enough cholesterol, your cell wall is really fluid and can break apart and rupture really readily, where if you have an adequate amount of cholesterol, it actually will rigidify that cell membrane and allow it to behave properly. So that embeds in cell membranes. And affects membrane fluidity. This is also something they typically check for at the doctor's office. So if you go to the doctor's office and they give you a blood test, they're typically looking at your cholesterol. There's good cholesterol and bad cholesterol. We're really not going to get into this in this class, but it's something interesting where if you take a nutrition class, you'll talk about it in more detail. Does that make sense? All right. I think we're going to just start amino acids, and then we won't get too far into it, and then tomorrow we'll finish up with DNA and RNA. But let's do a brief intro to amino acids, and then I might end class a little bit early. So amino acids, we already said, are building blocks, right? Specifically, they're building blocks for proteins. Does anybody know how many are naturally occurring? Yeah, it's in the 20s region. Typically, most textbooks will show 20. And these are referred to as amino acids. They're kind of the essential building blocks for making up proteins. All right, so let's take a look at the general structure. All right, the first thing that you have on one end is an NH2 group. And you've got an R pointing one direction, a hydrogen to the other, and an OH down here. Does anybody know the name of this functional group with the nitrogen? It's an amine. And then over here we've got an OH alpha carbonyl. What would that be? Carboxylic acid. Which is why this is called an amino acid, right? It's got both an amine and a carboxylic acid off of it. The interesting thing is that in your body, it doesn't actually exist in this form, at least not in a very high concentration, because we've got a carboxylic acid that wants to give up a proton. It's an acid, right? And what part of the molecule might accept a proton? It's not going to be the hydrogen. We've got an amine with a lone pair. Maybe that amine will grab that proton and steal it. So typically in the body, the amine will have an extra proton and carry a positive charge. And then this oxygen will have a negative charge. So this is under physiological pH, meaning in your body. And this has a unique name when you've got two charges, both a positive and a negative in a molecule. This is called a Zwitter ion. Where it's both positive and negative. Does that make sense?
So you only call it that when it's like occurring in your body?
Yeah, you only call it a Zwitter ion when it has both a positive and negative charge. If it is the one on the left-hand side where it's neutral and doesn't have charges, we don't call it a Zwitter ion. That's a unique thing about amino acids is you've got one end that's basic and one end that's acidic. And so you get these weird proton transfers that occur. If you go through your textbook, there's a bunch of these amino acids listed, and they're organized by groups. So you can kind of see the color coding system that they use. Typically, they organize them based on whether or not they're acidic, basic, whether or not they like water or don't like water. So this top group up here is hydrophobic. Does that mean they like water or don't like water? They don't like water, right? So if you think about a protein kind of twisting and folding up, these are typically going to be the amino acids that are on the inside of the protein. They don't want to be on the outside closest to the water. And then down here in the kind of teal region, these are hydrophilic, meaning that they like water. And when a protein kind of folds up into its own unique shape, these are typically the ones on the outside. You've also got in the middle these basic and acidic ones where depending on the pH, they will either be hydrophobic or hydrophilic, and that's variable depending on where they are in your body or where they are within a specific cell. But these are the 20 common ones that you'll see. You may notice too, I'm going to zoom in a little bit, that they're showing them with the oxygen with the negative charge and the nitrogen with the positive charge. So these are all shown as the Zwitter ionic forms of the amino acids. And over here, this is that R group that I had labeled before. So the R group for alanine would be a CH3 or a methyl group. And then oftentimes chemists and biochemists get really tired of writing out full amino acid names, so they use shortcuts. So for example, alanine can be written as ALA, where the first letter is a capital A, or you can just simply write it as an A. If we go down to valine, we've now got a different R group coming off of it, and this would be abbreviated as VAL or V. You may need to open up this chart when you're working on sapling. A few of the questions ask you to determine what an amino acid sequence is, and you may have to type in these codes. However, I'm not going to make you memorize these for the exam, so don't panic if you see questions like that. The other thing to remember too is that when you're looking at amino acids, they sometimes use weird nomenclature. So the end closest to the nitrogen is called your N-terminus. And the end closest to your carboxylic acid is your C-terminus. So you may see that nomenclature used occasionally in your textbook. It's basically referring to what direction you're looking at when you're going down a protein. Are you going from the N-terminus to the C-terminus, or the C-terminus to the N-terminus? And what we'll do tomorrow is we'll talk about polypeptides when we link multiple amino acids together. And again, that's a condensation reaction similar to what we covered yesterday. All right, so I think that's where we're going to end today. If you guys have any questions, though, feel free to stick around after class.